And I wanted to get started by actually just giving you an idea of the motivation behind the project and why we believe that's something which is really important to work on. So, I'm sure like you've all actually heard about antibiotic resistance and this being an issue, but to put it into numbers, right now there is more than like 1 million of accredited deaths, and the actual problem is that this is only expected to grow. So, I think something miraculous would need to happen for this to change, and we do expect more than 10 million deaths by 2050. And this, of course, is a massive global healthcare problem. But what really kind of, motivated me to work on this problem, and was like kind of like a really shocking moment, is that, if you look actually at the antibiotic pipeline and also antimicrobial pipeline, so things against, bacteria, we haven't really discovered the antibiotics for more than 30 years. And we aren't actually close to developing one. So, if you look at the scale of the problem, as well as the, the threat that it is right now, this really kind of is a call to everybody who, maybe would like to consider working on this, to work on such a problem. And there are two things which, I thought about when starting the problem, kind of two things that one could work on as a computational person. The first one is doing a better diagnosis. So essentially doing a faster diagnosis, which is more accurate. And the second thing was actually trying to help us understand the molecular mechanisms leading to resistance. So, what mutation actually drives the increase in resistance to a particular antibiotic? So the question that we kind of asked ourselves at the beginning was, all right, can we actually develop, a computational machine learning method that can help us firstly diagnose, but secondly help us to understand like the molecular mechanisms that lead to resistance. And this is something which could help us, for example, develop a new antibiotic or repurpose existing ones. If you look actually at the problem, each bacteria can be seen as a sequence of DNA. And this kind of DNA, it's essentially a code of life, and this defines what a bacterial cell kind of does. In a human cell, mammalian cell, it's a little bit harder. In a bacterial cell, it's actually a little bit simpler or significantly simpler. But what this kind of DNA does is that it tells us, for example, whether, or it defines the phenotype. And the phenotype may be that, a bacterial strain is actually susceptible to antibiotic. And this is, this is amazing. That's kind of what we want. But what often happens is that, bacteria, they keep mutating. They keep mutating, same as viruses keep mutating, and they're very, very fast at mutating. Kind of incomparably faster than a human cell because of how fast they actually kind of reproduce. So in a bacterial cell, what usually happens a lot, is that you have mutations. So you have a single nucleotide mutation, you have a deletion, or you have a disruption in a pattern in a transcription factor binding. And what this can lead to is this can lead to a change in the phenotype. So this can lead to actual antibiotic, not working. So the bacteria being resistant. And what I really want you to actually remember here is that if you look at this DNA, it's not often kind of like just a random sequence. It has a lot of patterns. So it's full of patterns like binding sites, codons, terminator, sequences. Anyway, a lot of patterns. That's what I want you to understand. And the kind of the goal of the model, the goal of what we wanted to do, is that we wanted to exploit these patterns to be able to understand how and why bacteria are resistant and how we can use that. So we've came up with a model called GeneBac, and GeneBac is essentially a deep learning method, because that's kind of my background, to try to predict antibiotic resistance, as well as can be applied to multiple applications. And over the next slides, I'm going to dive deeper into how it actually, what does it consist of, and why it works, and of course the results. So what it takes in is it takes in as input a DNA sequence. So it takes in a DNA sequence. This DNA sequence includes the mutations of the bacterial strain. So an example is a bacterial DNA. And we take kind of selected genes that we know kind of like are probably, you know, the culprit in this as the input, and we feed it through the CNN. And I'm going to talk more about the CNN, but essentially CNNs are really good at exploiting the patterns in the DNA. And what we do next is that because this CNN is actually able to capture the variation in the DNA, so see a mutation, reason from it, it can kind of lower it to low dimensional embedding. So it can output low dimensional embedding, which we then use as like to feed it into a GNN. And you know, why GNN? The reason for it is that really, if you look at any cell, a cell regulates itself. Genes in the cells, protein in the cell, keep interacting. So it really is like a very complex network. I would say such complex that we're very far away from modeling a single cell and how it works. So to kind of account for A, prior knowledge, and B, to be able to model these relationships between the genes, between the proteins, and here I mean genes and proteins quite interchangeably, we fit into a GNN, which we can then use to actually like predict the antibiotic resistance. So to predict the scalar value, which needs to be kind of, which you need to use of a particular drug to kill the bacteria. And as you can see, this is a pretty modular framework. That was the goal, because what is cool about it is that you can actually use it for multiple applications. So you can use it for antibiotic resistance prediction, but you can also use it for variant scoring, so to actually understand, you know, what are the mutations which are causing the resistance, as well as multiple others, which I'm going to talk about later on. And now I'm going to talk more about the convolutional neural networks. I'm talking to ML crowd, so I'm actually going to skip over it. But what I wanted to kind of just highlight is that, you know, CNNs are really good at exploiting patterns. So if you see this handsome cat right here, which is a cat of my friend, you know, this cat called Maciej has pretty, you know, specific face. And the CNN scans over the image and is able to exploit local pattern. So it's able to see whiskers, ears, eyes, and it can actually figure it out to, you know, output, let's say, a classification, which is here obviously a cute cat. And translating it to the DNA sequence, what the CNN does and why it is so good at kind of like processing the DNA is that it looks at the local patterns inside the DNA, how it is actually ordered, and is able to learn patterns like binding sites, like codons, and so on. So that's why we use a CNN. And how we call it, what the CNN learns is we call it kind of like the grammar, the DNA grammar. So we know, and this has been shown, it can learn kind of specific motifs. And also, I haven't explained what is a motif, sorry. A motif is like, let's say, a sequence of 10 nucleotides in a specific order. And it's very important that, you know, the model can learn motif specificity, the associations between the motifs, and so on. And something which I wanted to also say is that, you know, I'm not trying to reinvent the wheel here. It has been applied to multiple other kind of tasks in computational biology, worked well. Here, we just apply it to kind of, you know, pretty specific task. Okay, so CNN is done. Now the next one, which is the GNN. So as I said, genes interact with each other. If you actually look at a cell, you have genes which regulate other genes, which then regulate other genes, and some genes are helping other genes to regulate the genes. This is literally how it works. So to account for it in even a simplified way, we account for the interactions between genes by constructing a network using prior knowledge. And we're excited about it because we can account for the interaction between genes, B, we can also actually incorporate prior knowledge, and C, we can actually learn to, you know, weight, for example, these particular relationships, and see later on, you know, using something like interpretability, like GNN explainer, to look, okay, for a particular strain, what actual relationship was particularly important. And now I'm going to go more into the results and talk more about the, you know, whether it works, how it works, and how it can be used. So the first thing we asked was that, okay, can we actually try to predict antibiotic resistance based on the DNA sequence? And usually what was done is that, like, really the kind of state-of-the-art model is more using like a, for example, sequence of mutations, saying mutation is present, mutation is not present. So like a one-hot vector. And this is pretty boring because it cannot generalize to new mutations. And we know bacteria mutate pretty fast. And it cannot actually be kind of interpretable because, okay, there is a mutation, but why? Why is it so disruptive? Like, is it because it falls into, it reduces the protein stability? It falls into the transcription factor binding? So that's why we're really excited for using kind of DNA sequence model for it. And we took a data set of tuberculosis, of Mycobacterium tuberculosis, and we tried to predict essentially across drugs whether we can predict the MIC. And we can see that we're doing pretty well on it. And this is really cool. But what was actually kind of like the thing which made us excited is that if you look at multiple drugs, so on the x-axis you can see different drugs and performance, you can see that the model does actually quite well across drugs. And usually it was the case that the model can actually learn for 3 drugs, 4 drugs, 1 drug, but not for example, 9 drugs. And also you can see quite high variability. This is quite expected because in reality, all of these drugs have actually different number of training samples. So we could see that it can actually predict antibiotic resistance quite well. And the next stage was, okay, you know, can we actually discover the causal variants? So what actually kind of, you know, my supervisor but also biologists and specifically microbiologists are very excited are about, okay, tell me which particular mutation causes the resistance. So then maybe they can develop a drug for this mechanism, and maybe they can just understand kind of how the regulation works and how the resistance arises. So here what we did is we took a kind of database of mutations which have been already classified as, for example, resistant or susceptible. So causing essentially resistance or not causing resistance. And a lot of these mutations the model has not seen kind of at all during training. So it's pretty cool to actually see that the model, you know, is able to, you know, do very well and predict that the truly associated with resistance variants are actually causing kind of a change in their resistance compared to synonymous variants and variants which were not associated with resistance. And I then went to a couple of microbiologists and tried to actually kind of, you know, help us kind of do like in silico mutagenesis. So essentially mutate in silico the DNA sequence and try to uncover the variants and kind of highlight the variants which the model hasn't seen, or maybe we don't have the data for, but we believe could be resistant. And we actually, for example, discovered like a 1 missense variant which looks very kind of promising, and it falls into the right place. It disrupts kind of the binding system. It works on a specific gene. So we actually kind of knew that this what model should predict, but of course we haven't told it in any way. And last but not least, what is cool about DNA sequence models is they can model actually a range of mutations. So a mutation, mutations have different flavors. They can be single nucleotide changes, they can be deletions, they can be non-coding variants. And we compared it to like I would say the closest model available. And we did perform, you know, pretty well on all of them compared to the benchmark, which is kind of really exciting and shows that these DNA sequence models are pretty powerful, particularly for this problem. Okay, so third experiment out of 4. So often what we're interested in is we're finding genes which are causing the resistance. And genes often kind of like, it's often, not always, a result of, let's say, mutations in a single gene that the resistance, for example, arises. Because maybe this gene actually is way more active, or maybe more transcribed or less transcribed, and this actually affects the resistance very much. And if we knew the drug-resistant genes, you know, we could potentially repurpose existing drugs or create new ones. So what we did is we kind of run like a gradient saliency methods on the test set across a number of drugs and number of genes. So what you can see here is importance per drug slash per drug gene combination. And what we have seen is that if we looked actually at the existing knowledge, we see that we recover a number of genes that have been already shown. So we kind of know it from the previous knowledge that this is the case, but we haven't actually told to the model. So it's really nice that the model is able to discover it. But what is even more exciting is that, this is known, but can the model discover new ones? And this is something which we're like more working on. But the model also discovered a range of genes which we haven't really been kind of like discussed before in the previous knowledge, but can be potentially something that we can try in the lab. So we can knock out a gene, drug it, and see whether it actually affects the resistance. And the last thing I want to talk about is about transfer learning. So the problem in I would say biology as a whole, but specifically in microbiology, so for bacteria, is that you have a lot of data sets, but these data sets are really small. They're like 1,000 samples. And they're kind of split between, for example, different labs, and they're often of different modality. So the input can be the same, but the output is actually very different. The output can be resistance, or the output can be expression of a gene. And it would be really important to be able to kind of transfer learn, for example, across data modalities. And what we have done is we've taken a gene expression dataset from the Pseudomonas aeruginosa, so a really bad bug associated and causing cystic fibrosis. And what we've done is we've trained it on the gene expression task. So we've trained the CNN to predict the RNA expression level. And then what we have done is that we have transferred it to a model which predicts the antibiotic resistance. And the input in both is the same. And what we want from the model is also very similar, because we wanted to learn the features in the DNA. But this actually gives us a lot more training data. And what we have seen is that this kind of setup, where we train on the transcriptomic data set, then we transfer it to antibiotic resistance data set, does improve performance by, I think by a couple of percent. And what is actually even more exciting is that if you look at the improvement per drug across the data set, you can see that on the top right corner, it doesn't really, improve it. And that's expected. As these drugs, we have a lot of data, and often they have a well-known kind of mechanisms of resistance. But if you look slightly lower, you can see that we actually improve relatively to the kind of GeneBac without transfer learning and with transfer learning, showing that if you inject the model with prior knowledge in a way through transfer learning, it does better on the items, on the drugs, which it has less data for. So I'm going to go into conclusions. There's a couple of things I wanted to highlight, which we are all excited about. Firstly, that the model can predict antibiotic resistance. It can transfer learn across modalities. It can predict the variant effect, and it can recover genes. And I wanted to end kind of this talk with just kind of like because I personally really don't like kind of overselling papers and overselling, saying, okay, we've proposed something, it's solved. And, the reality is that we are very far from actually kind of like effective use in real life scenarios. And what we actually kind of need is in reality like just more data, like more high quality data, and investing into it so we can get into it, so we can actually get it. But the biggest problem in kind of microbiology and antibiotic resistance or antimicrobial resistance is that this problem is kind of like a global problem. So it's actually quite hard to find investors if it's a global problem rather than a actually local problem, like a problem of a country or of a region. And I would love to thank all of people who helped me in this project, including my supervisors, Andres and Sarah, as well as others, and particularly Aaron and Adam, who kind of like were really keen developing this project because I came in, I knew machine learning, I knew basically almost nothing like in biology, but I wanted to learn. And they took me in as bioinformaticians and said, okay, we're going to teach you this, you're going to teach us this. And it's really important if you work on this kind of interdisciplinary projects to have people who are willing to learn and willing to teach as well. And of course, the funders of this, AstraZeneca and G Research, who sponsored the grant to travel to this conference. Thank you very much.